So today we follow on after Jacques Miller's paper from 1961 with a paper by Max Cooper and collaborators Ray Peterson and Robert Goode. This paper was entitled Delineation of the Thymic and Bursal Lymphoid Systems in the Chicken and came out in Nature in 1965, actually in January of 1965. So it was really 1964. This paper became another classic in immunology. The background on Max Cooper is that he was just a young doctor, essentially, in 1965. He was from the South, from Mississippi originally. He went to Tulane Medical School and then did a internship and residency in pediatrics there. And then he migrated, the Great Migration. He migrated up to Minneapolis to have a research fellowship or a pediatric fellowship with Robert Goode. Robert Goode was a well-known pediatrician who was involved in little kids with immunodeficiencies, particularly. Max is the person who's been credited with all of the subsequent events after this paper, but Robert Goode was really the mentor in this whole process. Max was only 32 in 1965 when this paper came out, so he was just a young guy. He was learning the ropes, trying to figure out how to become a scientist because he'd been trained to be a physician. He wasn't trained to be a scientist, essentially. But in the introduction of this paper, the authors go on and they give some of the background in terms of why they did their experiments and what they were aiming at. Turns out that in 1952, there was a fellow by the name of Bruton who described little kids born with what was subsequently called sex-linked agammaglobulinemia. And these little kids didn't have any antibodies, and they didn't have any immunoglobulins, and they didn't have any plasma cells. On the other hand, they did have, they could generate what we call delayed type hypersensitivity, the cutaneous hypersensitivity that I talked about earlier, and they could reject skin grafts, and they had normal numbers of lymphocytes in the periphery, in the peripheral blood. So that set the stage for this. So that there was something, so that was telling people at this point in time is that whatever was responsible for the generation of the immune system, the antibodies and the beast and the plasma cells and so forth were part of it, but they weren't the whole part of the whole thing. And of course, then in 1956, a fellow by the name of Glick, who was a graduate student in the vet school at Ohio State University, accidentally discovered in chickens that if the chickens had their bursa taken out when they were neonates, just newborn chickens from the egg, ultimately they would not develop antibodies and immunoglobulins and so forth. And that was published in, I think I said the last time was in the Journal of Poultry Science, not a high-tech immunological journal. And so it took a while to filter down into other sciences and so forth. The bursa means in Latin, it means pocket. And what the bursa is sort of like an appendix in mammals, except it's an outpouching off the dorsal aspect of the colon in the chicken. And all birds have a bursa. And this bursa pocket is chock full of lymphoid follicles under the microscope. And it's named after a Renaissance physician professor from Padua, Italy, in the Italian Renaissance. It's called the Bursa of Fabricius. And he discovered a whole bunch of things. And so if you might want to go on and check him out, the Fabricius, because he's quite a celebrated type. They discovered in his papers, left after he died in 1621, that he had described the first person to describe this outpouching off of the dorsal aspect of the cloaca or the colon in birds. What happened with Glick at Ohio State in the mid-50s was that he was a TA, a teaching assistant, but he was a graduate student, and he needed chickens to immunize with something that he was using Salmonella typhimurium, typhoid, and so he needed some chickens. He wanted to make antibodies in the chickens so that he could demonstrate antibody activity and to his students. And he just borrowed some chickens that were in the facility that had been used by one of his buddies, another TA, who was studying the bursa and who had bursectomized these chickens when they were neonates, when I think they were only 12 days old. Lo and behold, they used about 9 of these chickens. 6 of them died when he injected them with, these were live salmonella organisms, and 3 of them survived. And when he looked for antibodies in the 3 that survived, there weren't any antibodies to salmonella in those chickens. And so that was his accidental discovery, but he went on from there and did controlled experiments and showed that there was a problem with chickens, bursectomized chickens, and their ability to make antibodies. So Goode and Max Cooper, said, well, this must be the model system to look at this whole thing. In the meantime, in the early 60s, they knew about Jacques Miller's experiments in the mouse, where he thymectomized the mouse. And it looked like that had an influence on the other aspect of the immune system. And so they thought, well, the experimental system to look in this instance is the chicken. They did similar experiments to what Miller had done in the mouse, with the added caveat is that after they did their surgery, either on the thymus or on the bursa, and removing those organs, they irradiated the mice to stop any more development of the immune system thereafter, to see what would happen. And that they don't go into why they did that, but obviously it made their experiments cleaner. There weren't these sort of gray zones. It was black and white essentially. And so in the materials and methods, what they go on to say is that from their experiments, and this is in the introduction, their interpretation of their results that you haven't heard yet, that I'll tell you about in a second, is that the thymus controls antigen recognition. This is the conclusion they made. They don't say why they concluded that, but that's what they concluded. Whereas the bursa controls antibody production. And of course, it's still at the time that cellular immunity and the cell-mediated immune reaction and so forth, allograft rejection, skin graft rejection. It was certainly described, from 53 on with Medawar, but antibodies were still the sine qua non of what the immune response was all about. So what they did was, in the materials and methods, was that they took two-day-old chickens. Now, an egg is not very big, and so a 2-day-old chicken is also not very big. But they operated on these, and they had a bunch of eggs, and they put them into 5 different groups. To one group, they bursectomized and then exoradiated. To another group, they thymectomized and exoradiated. And then another group, the third group, they did both a bursectomy and a thymectomy and then irradiated them. And then they had a 4th group there were chickens that weren't operated on, but just got exoradiation. And then a 5th group that were just normal controls. And so that was the experiment. So they did all these surgery on day 2 chicks. And then on day 40, so what, a month and a half, 6 weeks later, they injected these chickens, and now they're grown up some more, with antigens and to see if they could raise antibodies. One, they did 2 different antigens. They did bovine serum albumin and they did Brucella abortus organisms, microbes, bacteria. And then they waited 9 days after that. And they bled the chickens and then killed the chickens so that they could look at the histology of their different organ systems, the lymphoid system, and so forth. And that was basically this paper. And the thing is, they knew at this particular point in time that the spleen, which was a major organ that they could look at from the standpoint of, it's much bigger than the lymph nodes, and it sits in the left. In mammals, it sits in the left, in the right. Sorry about that. Upper thorax, upper abdomen, right under the rib cage, essentially. And the spleen is interesting because it has 2 different areas histologically that people knew about. And in the they had what they called the white pulp of the spleen. So because when you looked at it grossly, just if you just cut across the spleen and look at it, it has these white areas and then more darker areas and so forth. And when they looked at the thymectomized chickens, or the thymectomized and bursectomized chickens, the white pulp, the white areas had been wiped out. They were gone at 49 days, essentially. On the other hand, the bursectomized chickens had no lymphoid follicles. They were markedly decreased in the spleens and also in the lymph node. A follicle is just sort of grossly, it's a round collection of cells that you can see is delineated from the white pulp, essentially. There was also no plasma cells. They disappeared in the bursectomized chickens. When they looked for antibodies to their two immunizing antigens, the bursectomized chickens, whether or not they had thymectomy, didn't make any antibodies. So it's just like Bruton's agammaglobulinemia in the human, and which was freak of nature kind of thing. In the bursectomized chickens, when they looked at the thymus and the upper chest, the thymus was fine, no problem. And so that's the way it stood in the discussion or into the interpretation of these results. That's where they came to these conclusions, that the thymus plays a role in the generation of the antibody forming capacity, but it provides the antigen recognition. The immunologists use words from our language. And so recognition is, if you're walking down the street and somebody's coming towards you, and as they get closer, you recognize that's a friend of yours. You recognize that it's a friend of yours. So that's what the immune system does to antigens. It recognizes the antigens. Now, how they concluded that the T cells did that and the B cells didn't, or the B cells then somehow the T cells were communicating with the B cells to tell them to make the antibody, that was left up for conjecture. They go on to say that in mammals, this is in the discussion portion of their paper, they said, mammals must have a bursa equivalent. It must be the same thing in mammals. And at first they thought, well, there was and so over the next, this opened up an explosion of people, investigators, trying to find the mammalian bursal equivalent. And they first thought, well, it must be the appendix. It's an outpouching of the colon. It's got a lot of lymphoid follicles in it and other lymphocytes and so forth. And so they performed appendectomies on newborn rabbits and so forth. But they couldn't really, they basically took these poor animals apart from stem to stern looking for the mammalian equivalent. And that took place over the next 4 or 5 years. So this is between 1965 and 1970. And in 1970, some new things occurred. But that was sort of the frenzy of other people wanted to do what Max Cooper had done in the chicken, but they wanted to do it in mammals, because they figured that would be much more important and much more relevant for the human and so forth and so on, And then subsequently, some people thought that perhaps the tonsils were in it, because the Bruton's little kids didn't have any tonsils to speak of. So maybe they thought it was in the tonsils. And so they looked there and so forth and so on, never found anything. In 1965, 66, there was a publication in the Journal of Pediatrics by another pediatrician by the name of DeGeorge, who described a congenital abnormality where the little kids lacked the thymus. So they were and those little kids were very much like Miller's neonatally thymectomized mice. And the other thing that happened is that in 1966, and this is all subsequently to Max Cooper's paper, a mouse was discovered, a mutant mouse that was nude, didn't have any hair, didn't have any fur. And they're quite striking. They're cute little things. They're just hairless mice. And then a couple of years later, it took until 1968, when they finally looked. These mice were they didn't do so well. They seemed to be, they didn't grow as well as the normal mice, and so forth, and they died prematurely. They didn't really figure that out. But ultimately in 1968, it was found that these nude mice did not have any thymus. So they were just like Miller's thymectomized mice, and they didn't have it from even embryonic days, and then neonatal days after birth. There was no thymus, and so ultimately those mice went on to become workhorses in immunology, because they had a model system there that was just like the neonatally thymectomized mice. And so over this decade between 1960 and 1970, it became evident, because of Miller's work, because of Cooper's work, and because of these mice and the congenital abnormalities in the human, that there were two different immune systems. There was one immune system that was responsible for the generation of these lymphoid follicles in the spleen and in the lymph nodes, and then subsequently the maturation of lymphocytes into plasma cells and the ability to make antibodies. And then there was this other system that were the lymphocytes, small round lymphocytes with not much cytoplasm, that arose in the thymus and then seemed to populate the periphery throughout the first weeks and months of life, to the point when after in the mouse, after the same way in the human, after puberty, then the thymus involutes. But now you've got lymphocytes all over the body and in the blood, in the lymph nodes, and the spleen. And those are the cells then that ultimately were found to mediate graft rejection and are responsible for the phenomenon of cell-mediated immunity. So those are the 60s. And as you recall, I started the 60s with Peter Noel's paper where he was the cytogeneticist looking for a way to separate red cells from white cells, and found that phytohemagglutinin, this crazy plant protein, would cause the lymphocytes to proliferate. Well, that then also figured in, because people started doing what Peter Noel had doing, and suddenly over the course of the whole decade, all kinds of people came into this new field of cellular immunology, and created a whole explosion of new information about how the immune system works. So I'll stop there. And if you like this journal club, please say you like it by clicking on the like button. Go to my website, kendallasmith.com, and scroll down on the homepage down at the bottom. Sign up for my newsletter, because that's where I'm serializing my new book, The Quest for New Knowledge, that you might find interesting in terms of how science occurs, how science gets done, and so forth. And on the YouTube site, like it and subscribe, because we need to generate more people who really like immunology and want to promote it. And we need it right now, because immunity is really wonderful if you've been vaccinated recently. So I'll stop there.